So for our first speaker today, I'd like to introduce Kamil Jaron, who is a postdoctoral research associate at the University of Edinburgh. And Kamil is going to tell us about some really exciting work that he's been working on, characterizing features of polyploid genomes from sequencing reads. So many of you have seen almost certainly his work with Smudgeplot, and I'm really excited to hear him go into the details of how all of this works. With that, let him take it away. Thank you.
Oh, thanks a lot, Mike. So, and thank you all for participating. It's very exciting to actually open this amazing seminar series. So, I will start right away with the problem that is very well known to all of us, that, well, when we assemble a genome, well, where we actually sequence the genome, what we actually ended up with this huge pile of reads, and sometimes it's more difficult to put it together than one might like. And that's why we wanted to advocate for approaches that are somehow helping us to expect something from genome assembly, some approaches that allow us directly to sort of see what we have sequenced. And so what's the problem with the assembly? Well, if we start with a simpler case than a polyploid genome, but with a simple diploid genome, it just might happen that there are some duplicates and there might be some heterozygous regions. And if we just naively assemble it and the assembler is lumping together everything under a certain similarity and separating something with a certain divergence, we might happen to assemble the heterozygous region separately, or we might happen to collapse too many paralogs. And this is not such a huge deal if we at least are able to quantify this, but we just don't know. Like, when we assemble genome, it's really hard to say whether it makes sense what we have done or not. So this problem even blows for polyploid genomes, right? So more genome copies are there, more heterozygous types, like sort of conformations that exist, and it just generates more and more problems. And we would like to get at least some other prior information before we start with processes as complicated as genome assembly. So it's great to have genome size estimates via flow cytometry. It's great to have karyotypes and know what's your ploidy, but it's not always available. And these techniques, although they are not that super difficult, there are some systems where they might be quite sneaky. So there are also a lot of techniques that are operating directly on the raw sequencing reads, and these are techniques I'm going to talk about today. There are approaches how to analyze individual genomes and some features of individual genomes, and there are also some quite recent tools for population genomic scale Kmer analysis, which I find pretty cool, that where they do a GWAS directly on Kmers. This is something which is not the aim of this talk, but I do provide some references in the supplement if you're interested about this. I will talk about the estimates of genome size, heterozygosity, and ploidy. So what's the point of decomposing, like what is actually Kmer spectral analysis? Well, the idea is that you take a read and you take all possible subsequences of certain length K. That's why they call them Kmers. So in this example, we have K of that's equal to 7, which is sort of unrealistic for most of the bioinformatics Kmer analysis. That usually we use Kmer lengths that are much longer than that. It's like 21 to 35, let's say. However, I just wanted to show you in this example that decomposing read to Kmer means taking first to kth or 7th nucleotide in this example, second to 8th, etc. So every single read produces us the read length minus the Kmer length sub-Kmers. So what's the point of, isn't this just throwing away a lot of positional information? Well, maybe it is in a way, but if you have a genome and you sequence it with, there is a certain error rate, right? And even if you have Illumina sequencing with error rate lower as, I don't know, 0.5% or 1%, it just happens that most of the reads, or like loads of the reads, actually do have an error, which are here marked with the X's. And what you would prefer, if you would like to do like any statistics on this, you would like to be able to separate what is an error and what is not. So the idea is that if you decompose a read in all the possible Kmers, only the Kmers that are around the sequencing error will inherit the error, while lots of other Kmers from the rest of the read get rescued. So actually the signal to noise ratio improves a lot. And then what we can do is just for every single Kmer, estimate or calculate the coverage. And we immediately start to see that some of the Kmers will have coverage that is alike, while some other Kmers will have very low coverage. So these low coverage Kmers are most likely errors, those that are around a single mode. These will be our real genomic Kmers. And there are obviously some Kmers there will be somewhere in between that are hard to say, but we wouldn't be trying to make any claims about any particular locus. We will try to fit their distributions. That's why it's sort of okay. So if we plot a histogram of the coverages, it might look like this. So again, I started with a diploid because let's say it's simpler. And I also picked a nearly completely homozygous genome, or maybe even a completely homozygous genome. So all the Kmers that are genomic are in 2 copies. So this is the main peak. We see that there are a bunch of Kmers that are with low frequencies. These are our sequencing errors. And we also have this small lump that is about approximately twice the coverage of 2n peak, which represent genomic duplications. And the histogram is obviously tourniqueted because there will be a lot of repetitive sequences all the way right. So if we add heterozygosity to a diploid Kmer spectra, we just get a second bump over here. So these heterozygous Kmers are in a single genomic copy, while the homozygous Kmers are those that are in a double coverage, so in 2n copies, and then again we still have the duplication bump, etc. And so you can kind of already see that comparing the size of the heterozygous Kmer distribution versus homozygous Kmer distribution can give us some insights about what's the heterozygosity of the genome. And this was done. It's actually there are a bunch of implementations for this, but I would say that one of the recent popular ones is GenomeScope, where the model fits 1n coverage, and basically this, given the 1n coverage that was also fit as a parameter of the model, it estimates the genome length, heterozygosity, and repetitiveness. It's also a model that is aware of the duplications, so it sort of assumes that the heterozygosity might overlap with duplication. So there is a bit of combinatorics involved, but it's not like that insane to imagine how it works. So this is not that easy for polyploid genomes, and luckily we have smart people like Riker who are actually good in combinatorics and are good in figuring this stuff out. So I actually had a bunch of polyploid genomes, and I was interested in characterizing their heterozygosity, and that was the moment where I was contacting authors of the original GenomeScope paper, and they were saying, hey, we have a polyploidy tool already ready, and it was really great. So we started to put together my polyploid genomes with their tool, and we've done a lot of genome profiling together. And the idea is that I will show you a triploid case. Wait, sorry, I didn't see half of my slides. So instead of 2 peaks, we have 3 peaks. The first peak, again, there is a 1n, second peak is 2n, but we also have the 3n peak. And the Kmers that are belonging to these 3 peaks are basically of 3 different types. So we have a homozygous Kmer, so those that are same between 3 loci in the triploid genome. So they are in 3n copies. We have heterozygous Kmers that are biallelic. So that would have 1 Kmer that is haploid or monoploid, 1 Kmer that is diploid, so 1 and 2n, and then we have also loci that are 3 allelic, that have 3 different states in all 3 genomic copies, and these will just generate 3 different Kmers that will contribute to this first peak. So we can again compare the first and second peak to get the amount of biallelic loci, then estimate the rest of the 1n peak to get how many 3 allelic loci we have, And then we have also the homozygous Kmers, which give us a comparison to the two, and we can sort of profile it. So if we fit the model, we get an estimate of monoploid genome length. We get a fraction of genome that is unique. We have the estimates of the heterozygous loci that are AAB type and loci that are ABC type. We also have the estimate of the coverage. So it's really great. And in this kind of case, you don't really need much more than seeing that you have 3 peaks. But we also have seen a bunch of Kmer spectra that look like this. And we were like, is it like a triploid with duplications or diploid with duplications? Or is it like a tetraploid? Which one it is? It was really hard to tell. And then we thought, okay, is there any information, the Kmer spectra, we just didn't utilize yet? And we thought, well, okay, if it's a tetraploid genome, we might expect to find some of the Kmers that are in a 2n peak pairing up with the 2n peak as well, because the tetraploid genome will have heterozygous size that will be either AABB or AAAB type. And also we would find some Kmers that will be pairing from this distribution to this distribution. So we had this idea that we cut all the error Kmers out. We cut everything that is in high copy number because we are trying to understand the core of the genome. And then we search for all the Kmer pairs that are different by a single SNP. This is a purely computational requirement because this is something we can actually do on a huge number of Kmers. When we would start consider also indels and things like this, it would get computationally really complicated. So we've decided to start simple, start with a single SNP, and it kind of worked out quite well. And we have a second requirement that every single Kmer that we find must be a unique pair. So if we take an example, now my K is 5, which is completely ridiculous for any type of analysis, but just for illustration of what I call a Kmer pair, the first 3, although they are 1 SNP away from each other, they don't form a unique pair. There are 3 Kmers that are 1 SNP away from each other, so they would not be considered in the analysis. While the 4th and 5th Kmer, they are 1 SNP away and they form a unique pair. That's why they would be included, etc. And we sort of hope that these Kmers are mostly representing 2 alleles of the same locus, so the heterozygous Kmers, which, if you're thinking that does not have to be case, you're right. So then we do 2 transformations. We do a transformation of summing the coverages, which gives us an idea about how many genomic copies are there of this Kmer pair. And then we also look at the proportion of the this should be proportion of the coverage of the less represented allele. So it's coverage of the less represented divided by sum of both. How does that work like? Well, if my 1n coverage is 100 in this example, the AB type of loci will have approximately 200x, and the coverage, I will call it the coverage ratio, although it's not exactly ratio, but it's around 0.5. Then 3 like triploid loci will have 300x coverage, etc. And the coverage ratio of AAB will be 1/3 because it's like B divided by A plus B, which is 3. Which is great because if we then plot it on a plane, the coverage sum versus coverage ratio, it sort of like spreads the genome structure in a 2D plane. So we can just look at the intensities of what we call smudges. That's why we call it the Smudgeplot. So how does a Smudgeplot of the Kmer spectra you have seen looks like? It looks like this. So we actually see that we have 34%. Ooh. What's that? Well, sorry. We have 34% of our Kmer pairs belonging to AAAB smudge. And we have also loads of other Kmers there too. But basically we say, well, it's probably tetraploid because it's sort of here is the cut of where we see a lot. For example, we don't see the tetraploid peak from the histogram pairing with anything else. So if we would sort of put this together. By the way, this is a root node nematode. This is a melodigene urnaria. So we see that the AB smudge, which represents a Kmer from this distribution pairing with itself. We have AAB smudge, which is pairing of 1n and 2n distributions. Then we have AAB, which is the 3n, 1n, and then we have AABB, which is 2 with itself. Yeah, I forgot to say that these marginal histograms on the sides are just like sums of rows and columns. So basically, this is just a distribution of the minor Kmer coverage, and this is a distribution of total coverage of the Kmer pair. So, all right, this is the idea. Now I will show you a few applications. They had this like really cool system of root node nematodes, and one of them is really peculiar. It's melodigene floridensis, and it's peculiar because it's a diploid among triploid or tetraploid species. And basically it's a quite big clade with lots of root node nematodes that are a product of hybridization. They've done a lot of work to really make sure that this melodigene floridensis is actually part of this clade somewhere in the middle. And it really is. So it was not clear whether they have like so many acquisitions of polyploidy or actually a reduction of polyploidy in a diploidy, which is sort of like it was an interesting system. However, although the melodigene floridensis is very different to all other root node nematodes in the clade, it's actually a triploid. So how comes that they just didn't figure out that it's a triploid? Well, what had gone wrong? The thing is, if you look at the Smudgeplot, it just like has this super bright AAB smudge, so we sort of think it's quite a reliable triploid. But if we compare it to a different species within the same clade, a different triploid species, melodigene incognita, then the Kmer spectra looks very different. While the incognita has a lot of AAB and ABC type of loci, the floridensis is much more AAB based. So what is the deal with it? Well, it seems that the incognita has 3 different genomic copies that are divergent. So when they took the sequencing reads and assembled it, sometimes they recovered all 3 genomic copies from the genome. While the floridensis that have 2 subgenomes that are extremely similar never had this property. When they assembled the genome, they found the genes at most at 2 copies. That's why they thought that it's diploid. And there is also some cytological evidence from before that it's diploid. But apparently what they have sequenced is not. Well, so it like there is no wonder that there was this confusion. It really does look like a very different genome. It's just still triploid. So what happens if the genomes are not very heterozygous? If they're homozygous? Well, I have this example of parasitoid wasp. And if you look at the Kmer spectra, it fits a model. It has a good estimates of the genome size that's approximately accurate. What's not that cool is that it still estimates some heterozygosity. And from the biology of the species, we know that there is none because it's a species that's reproducing with gamete duplication. So there is every single individual starts as a haploid and duplicates in a diploid. So this is sort of an artifact of a sort of skewed sequencing coverage and the model fitting there. So careful about this when you have a very low heterozygosity genome. Well, and what happens if you run a Smudgeplot on this one? Well, it actually moves all the labels 1 level down. So it thinks that the 1n coverage is 68 because all it can do is to pair up the Kmers from this distribution with itself, but it's wrong. So actually, if you know that there are no heterozygous loci and you tell Smudgeplot that is the case, it can label you correctly the smudges and sort of estimate you correctly the 1n coverage that is in the 31x. But it's a trap. Like if there are no heterozygous loci at all, the Smudgeplot gets it wrong always. But what we're looking at then, if not heterozygous loci? Well, it's just simply the paralog structure, which I also find cool, but it's not what we were looking for in the first place. So I sort of assumed that in Polyploidy seminar there will be mostly plant people, and I think all of you will love strawberries. So I actually, when I was like figuring out where are the limits of Smudgeplot, did run the octoploid strawberry, and I was very pleased that actually I did estimate it as octoploid, and I actually the Smudgeplot figures out there are quite a lot of loci that have like 6 copies the same and 2 copies different, which is sort of consistent with a lot of genomic analysis that came out later on, which I was very pleased about. However, it was not always the case. So now we have finally cases where Smudgeplot really gets stuff wrong. And that's when the duplications are dominating the heterozygosity signal. This is a case of a different strawberry that is known diploid, because they've done lots of work on these strawberries. So they actually know quite well. And the problem is that the heterozygosity peak is very small, and there is actually quite a decent duplication peak. And it just happened that we pick up more duplication Kmer pairs than heterozygous heterozygous Kmer pairs, which ends up that we estimate as a tetraploid. I actually don't really know how to deal with it so far, but if you look both at the Smudgeplot and the GenomeScope, usually you can figure it out, which is actually great. I'm quite grateful for the pairing of the two, which is nice. So now I would just like to, I'm running out of time. So there's just a few considerations. So GenomeScope works great, but it does require, or like this, it does assume a uniform distribution of heterozygous loci and duplications. So, which does not have to be always met. If the fits the one uncoverage somewhere else, it will get you all the numbers wrong. And also it cannot deal with super huge heterozygosities because there are just no shared Kmers. However, it has negligible computational time, it scales to any size data set, and you can actually fit quite good models in quite modest coverage. And it can have a really accurate genome size estimates. And I don't have time to go through it, but I have a bunch of blog posts about it, and there is one really cool paper, and I linked it in the supplement. Smudgeplot has this big weakness of requiring heterozygosity dominating paralogy, and it does require a bit more coverage. Computation is still rather exhaustive. The biggest genome we've done so far was 3.5 gigs get deployed. So sorry for this. I guess it won't satisfy a lot of plant research. However, it's a descriptive visualization tool. It does not lie about your data. It's completely honest, and it pairs up very well with GenomeScope too. And it actually does a very decent one-n coverage estimate. So this is not to complain about genome assemblies. They are still useful and great, but the whole point is just to have a big scale picture before anything as complicated as genomes assembly. So if you are interested in like looking at weird genomes and have something to share, I'm actually running a database of peculiar genomic observations. So if you would like to contribute, I would be super happy to incorporate it. And with this, I would like to end up and thank a lot my collaborators that gave me this most amazing experience, and they were actually the reason why I started with all this Kmer madness. So big shout out to Riker and Mike. And these are just the lots of links to different resources about Kmers. These are the references of all the figures that were used in this presentation. And with this, I would like to thank you all for your attention.
Thank you.
Yeah,
that was wonderful. So as we try to navigate this first chat or question session, real quick, we have a few minutes for questions. If people have questions, let's just tap them into the chat, and then that'll give us some way for people to raise their hands, because there's 230 people on the call. It's a little bit hard to see everyone's screen at once. And then you can go to video and audio and ask your question as that. So who has a question for Kamil? All right, Alyssa, go for it. I'll let you ask your question.
What level of coverage is required, and how do you think required coverage will increase as ploidy increases?
So this is a very fair question. The coverage, the smallest coverage we successfully ran GenomeScope with was like 15x per monoploid copy. However, this does need like a really good sequencing run. Actually, we don't talk about this much, but if you have like different Illumina libraries, sometimes they have an order of magnitude different dispersal in coverage variation. And bigger coverage variation it is, more coverage you need, because it's just harder to distinguish the individual peaks. So I would say 15x per monoploid copy if you're lucky. If you have 50x, it's great. If you have 100x, you're safe. Okay, I will go them one by one. Can you talk more about computational needs to run both programs? All right. GenomeScope is entirely like it's calculating the Kmers is on lock count. It's like it doesn't matter how big is your data set. The KMC or a different like Kmer counters will do it without any problem. And running the model itself is also a few seconds on your laptop. So that's without anything. What is quite expensive is to find the Kmer pairs. We have a C++ implementation. But it still works on a single core, and it does require a lot of memory. So the bottleneck is that I think it's 20 times the size of the dump file. So if you have 20 gigs of Kmers, which is not that a problem for a plant genome, you will end up with 400 gigs of memory in your computer. So for plant genomes, I think the biggest issue is the memory. Now over time, both reads. Do you think method would work with error-prone long reads? So I've seen one run with long reads and didn't work at all, but I have not seen the histogram. I've seen just Smudgeplot, and I think I would like to explore it a bit further before I will have a definitive answer. But so far, I would say no. There are just too many indels in the Kmers of long reads for this statistical analysis to work. Have you tried using non-Illumina data? So we tried. I've seen 10x, which is still Illumina based, but that worked out. I'd never tried a nanopore.
It doesn't work for long reads.
Sorry, say again.
So it doesn't work for long read data.
Yeah. So I've seen a run with error-corrected SQL run, and that didn't work at all. Like there was just basically there were too many error Kmers with high frequency that they were just pairing up with the real genomic Kmers and sort of made it like one huge smear over the whole Smudgeplot.
So I tried to use GenomeScope for Kmer generated from long read, but it doesn't give you the correct assessment. But
okay,
portrait it gives you somewhat reliable thing, but long read is just a messy estimate actually.
Yeah, so if you look at just the Kmer spectra, do you see the genomic peaks, or does it look more like just an exponential decrease of coverage?
It's just kind of exponential decrease of coverage, I would say.
Yeah, so this is what I would expect. Like it's really difficult with error-prone reads to have this kind of like nicely shaped Kmer spectra.
Sorry, I just missed the first 15 minutes, and so what kind of data are we just used to use the SmartJet, or
so
it only work because I just missed the first part, the most important part. So yeah.
So I think so. Right now we're running it only with Illumina data, and there are lots of other questions, and I can try to answer them in chat, but I think we're running out of time for my slot.
I think go ahead and you can ask. There's a lot of folks who I think want to know the sort of answers to George's and Tyler's questions there about diploidization and distinguishing sort of the nature of ancient polyploid events, as well as if you could use low coverage data or even subgenomic data like RADseq. So go ahead and address those, and we'll get have you stop sharing your screen and let Michael McKain get ready How's that? No, you're good. So yeah, feel free to answer those last couple of questions.
Yeah, so the signal over the diploidization process, could it accurately guess the policy of a mesopolyploid? Wait, that's I'm lacking vocabulary here. Well, mesopolyploid is the one that has the still tetraploid structure, but it has a preferential diploid pairing. Well, so I think it's always the function of like, what's the divergence between the copies? If there are still shared Kmers, we can pick them up. If there are no shared Kmers between them, then the signal is gradually lost. How fast it decays, that's really, really difficult for me to answer. I think we would need to see a bit more data sets. I think there is an Arabidopsis data set that could be used for this. I'm not sure, but I have not explicitly tested this. But I would be very keen to figure that out. Chad, any thoughts? So I've seen a DD RADSEC, and it didn't work very well because the RADSEC has a lot of amplification steps, and they also increase a lot the coverage variation. So basically on a Smudgeplot, you can imagine that it smears all the smudges vertically, and well, actually also horizontally, which makes them very much harder to distinguish. So you just end up with completely like one huge smudge. So I just wonder, like in theory, it should be possible if you have sufficient coverage that because the coverage variation is random, so it should average out. But I haven't seen a successful RADSEC Kmer's Smudgeplot, especially in theory about the regions between different distribution. I'm just thinking this. Yeah, I don't know. Rob, yeah. So if people have like DD RAD data and they believe that it has like a reasonable coverage variation, I would be very happy to give it a try. I'm just skeptical about anything that went through PCR.
All right.
Great.
Well,
okay.
Thank you, Kamil. All right. Well, let's hand it over to Michael McKain, who just had his slides up there looking good. And we'll let Michael get those fired back up again. So Michael is an assistant professor at the University of Alabama, as well as the curator of the herbarium. And today he's going to tell us about the impacts of homoeologous exchange on polyploid subgenomes. And with that, I'll let Michael take it away.
All right, thanks, Mike, for putting this on. This is actually a lot of fun. I was practicing a while ago, and I realized how bad my oration ability has gotten since I haven't been talking in front of people for a month. It's just me and my 11-year-old daughter hanging out most of the time. So we'll see how it goes. Luckily, I've done a lot of this before. So if you've seen my talk, like basically the talk I've been on the circuit with in the last few months, is kind of a short version of it, but I still think this is a pretty interesting topic and something I think as we all work on polyploid genomes, we should really start to think about more the impacts they might be having on our data, especially those of us who are doing like phylogenomics with polyploid genomes. I'm just really curious to see, as we add more data sets to these type of analysis and think of them in this way, what results, what's kind of going to shape out. You know, I'm a big fan of the belief that lineage specific stuff is just everywhere. So, you know, big rules that kind of guide things are kind of hard to come by, but just say, oh, it's a lineage specific thing and you'll be safe. So what I'm going to talk about is this homoeologous exchange and its impact on our understanding of polyploid subgenomes. So since this is a polyploid seminar, I figured I'd just jump right into talking about genomic consequences of polyploidy, since we all assume polyploidy is really important, amazing, and awesome. So the first thing to remember is that there's usually, or not always, but often a potential restriction for gene flow. So this is simply like distinction between a polyploid generation and a diploid generation. Sometimes it's hard to cross back, or you get the triploid bridge or something like that. So there might be instantaneous distinguishing between generations and ability to mate. We see increased chromosome number and genome size. Again, this is kind of what you would expect if you're increasing, adding two genomes together with that reduction. Makes sense, right? Increase the chromosome number, increase the total genome size. This isn't necessarily always double. There's a lot of variation that goes into this. We see a lot of loss of chromosomes and things like that, but still it's occurring. Something I think a lot of us talk about and think about are just the gene copies. So like, what are those gene copies doing? How many are there? How are they turned on, off? Like, what are the interactions going on? And I think that the most interesting aspect of this is simply that, you know, though we talk about copies a lot, remembering that those copies are parts of networks, and those networks are now duplicated, and the interactions within those networks can change, and potentially might be one of the potentially coolest parts of like the diploidization process. And that leads me to the last part here, is just this idea of diploidization. So this concept of moving from polyploid-like to more diploid-like through the fractionation, so loss of different paralogs between paralog pairs, changes in expression of variation between subgenomes and paralogs, methylation repatterning that seems to come immediately pretty much after the polyploidization process, like variation and expansion and contraction of transposable elements from either subgenome or across subgenomes. And then the topic that I'm interested in talking about today, which is homoeologous exchanges and the associated genome rearrangement, that I'm going to focus more on homoeologous exchange from a perspective of phylogeny. So what do we mean by this? Well, imagine that you have a polyploid event that came together from species A and B, where species A is purple, species B is green. And then over time, we have homoeologous exchange occur. So over time, we start to see that these are just kind of like translocations occurring. They're moving bits and pieces across the subgenomes. We know this is occurring because we see, you know, our mesopolyploids or our ancient polyploids all mixed up when we look at their synteny and their genomes. And then sometimes this can result in things where it's not exactly, you know, perfect translocations of copies, but we get variations in copy number. So you might have 3 copies of the green genome and 1 copy of the purple genome or something like that. So when we go and we interpret this, and we've seen this interpreted different ways, remembering that having genomes on hand is still fairly new. You know, when I was in grad school, the idea of doing a genome as part of your dissertation was ridiculous almost. And now it's like, oh, it's chapter 1, right? Or multiple genomes are chapter 1 for grad students. And so, you know, when we looked at this data, or we look at this this concept and think about all the data we have in hand, we have a lot of shotgun kind of data, right? We have this ability to, or maybe a lot of SNP data or something like that. So we can come in and we can think about, well, if we were interpreting homoeologous exchange and the possible true nature of what this is and what it looks like to us, we can see different things. This is coming from Scheisel et al. In 2019, where if we have reciprocal translocation, so we're doing this down here, we have A1 and B1, they just flip-flop, right? They just translocate. So what was on B1 is now on A1 and vice versa. Synteny remains the same. Genes are the same. There's no change other than that. They're just moved location. We can have something that's deemed non-reciprocal translocation. So in this case, instead of A1 and B1's genes 1, 2, 3, 4 are, instead of just translocated and switched, what we see is like copying over. Sometimes we see terms like gene conversion, or we use homoeologous exchange with replacement or something like that. You know, there's a lot of different words kind of describing very similar things, at least in some ways. But in this case, if you were to look at just like shotgun data or a SNP data or something like this, this would look like a presence-absence variation, right? Because instead of having the alleles that we had before, now we've just got 4 alleles of the same thing, or it's homozygous across all sites or something like that, or which to us might read, oh, we lost a copy, like lost one of the paralogs. And then you can see the same thing kind of happen here, similar but different, where we have translocation heterozygote. So instead of everything's all one way or the other, it's kind of mixed. So in this case, instead of having 2 B1s and 2 A1s of the 5, 6, 7 string of genes, syntenic block, we see that there are 3 A1s and 1 B1. So what we would interpret that as a copy number variation. So we would see a change in copy of maybe A1 or something like that. So just keeping in mind that the way we interpret this, you know, when I'm drawing these pictures, or when Pat and I drew these pictures for this paper, we were thinking of like whole genome, when in reality, like a lot of the time we don't have a whole genome, especially a chromosome level assembly of a genome. We simply have a lot of reads or something like that, or maybe we just have a lot of SNP data. But if we kind of look at what other people have done, we can see that homoeologous exchanges, so this idea of like moving things around across homeologs, is prevalent, especially when we look at it in ways of maybe like resynthesizing polyploids. So, you know, Brassica Nappas being like one of these amazing systems with all these resynthesized polyploids, and just a really cool system that sometimes I get jealous and wish I worked in, but then I'm like, okay, there's a lot of great people working in it, I think they're fine. But This paper from Higgins et al., they demonstrated that when they resynthesized all these lines and they looked at homeologous exchange, they saw that it varied. So basically, if you see color changes here in this like these gray boxes, this equates to some type of homeologous exchange. There's some variation in that, but what you'll also notice that there's certain chromosomes that tend to have quite a bit repeated across multiple resynthesized lines, suggesting that there might be some aspect of this that's not just stochastic, but there's some inherent thing going on that might promote homeologous exchange. You can imagine if it's like maybe 3D structure within the chromosome or something, or within the nucleus or something like that. So they're just 3D rendering them together with chromatin, and they're just spatially close to each other, and then might cross over. What's cool is we've seen this genome restructuring in other organisms too. I'm going to be all plants today because, you know, it's my bag. So Kamil, you were right. It's all plants, at least for me. So Brassica napus being one, Jalcipium, so cotton genome, Arachis, so this is peanut, which is a tetraploid, Triticum, so the wheat complex and all its really cool stuff, and then Tragopogon have all demonstrated something like this. Not to the extent that Brassica napus has been studied, but all have some type of genome restructuring. It seems to be suggested by homeologous exchange. So what does that mean from another perspective? So when we have a polyploid genome that's kind of coming together and you know, working it out and, you know, functioning, has to be some resolution of this subgenomic conflict. You know, these two subgenomes functioned just fine on their own, and now they have to come together and function as a whole. There's a lot of changes that occur really rapidly to do this. You know, and this idea of subgenomic conflict, of whether or not these subgenomes are on or off, or portions of them are on and off, or what's happening, you know, we can have this idea of extremes where one subgenome, so in this case, the red subgenome maybe dominates over the blue, or vice versa, blue over the red. We can have a situation in which they just kind of mix their contributions across the whole body of the plant. So maybe those gene networks had 5 genes in them, and it's just one from the subgenome back and forth, you know, back and forth or something like that. Or you can have something where, maybe it's a separation by state, so vegetative versus reproductive. So in this case, the vegetative material is supported by the blue subgenome, and the red is supported by, or the reproductive is supported by the red subgenome. So all this is kind of happening, all getting figured out. But when we have homeologous exchange, especially with replacement, so we're changing the history of we're changing the copy to one of the other parent's copy of the genome, then that just kind of throws a wrench into everything, right? Because everything was worked out, and now it's got to rework itself out. And we've looked at, when people have looked at this, so Lloyd et al. In 2017 looked at this, and they found that prior to homeologous exchange, you can have expression variation between homologs. We expect that, you know, one subgenome might be lower expressed than the other, pretty typical. So what happens then if you overwrite the lower expressed one with the higher expressed one, or vice versa? So now you've got 2 subgenome like paralogs that might have been expressing different levels, and you're still trying to make them work in the system. So this expression post homeologous exchange can be affected by which copy is maintained. It can be additive, and they can both go up, they can both go down, they can change. I can also imagine it depends on what portions of the genes were actually copied over. So you might have a different gene, like the different vexon space, but maybe you didn't copy over the regulatory region. So it's the same transcription binding factor sites and stuff like that. So maybe it doesn't actually, it still has like a similar pattern to what it had before for expression. These are all possibilities, right? But what we kind of expect and think is that over time, you're going to see that this expression will eventually, the copies will kind of work themselves back out, and that will all reflect dosage needs, which is kind of like the name of the game, right, for polyploid expression, dosage. So if we want to investigate these things, what we really need to be able to do is reconcile subgenomes to some type of identity. So I think in the last 10 years, you know, I know Mike's lab, I've worked on a lot of this, lots of people have been working on just identifying polyploid events and where they occurred. And now, you know, I think we're getting pretty decent at that, right? So now it's kind of coming in and say, okay, how do we tease these together apart? Can we answer really interesting questions about subgenomes and these type of questions about expression variation, maybe homeologous exchange, but we need to understand which subgenome these originally came from. So, you know, from the phylogeny perspective of polyploidy, you know, this is like your easy example, right? Where we had a phylogeny of diploids, so A, B, C, D, E, F, and then A and B made a polyploid together. And then they diversified after that. So there are 8 AB species or 7 AB species. But within, if we looked in those AB species, we would see that there's a copy of a paralog that's very similar to B and a copy that's very similar to A. Now this is a very clean, like all things working phylogeny of a polyploid. And we all know this is garbage for the most part. And if you actually do this and look at them, just take a shotgun and randomly cut out things, and its interpretation takes a little bit sometimes. So just in general, loss and fractionation is a problem. But then what happens if we throw homeologous exchange, especially with replacement, into the mix? Well, then we have to we rethink those histories, because just because there's 2 copies doesn't mean they're the original 2 copies, right? So you can imagine this. So we have our AB species come together, make our nice polyploid. You know, this is what the phylogeny would look like. It's a nice little network. But if we were looking at these gene trees of interest, so we'll do something simple. We have A, B, C, and then we have A, B. So if there's no homeologous exchange, so the chromosomes are the way they were, they're just in the subgenome, we would have something simple. 1 copy is related to A, 1 copy is related to B. Exactly what we would expect. If we have homeologous exchange with just a translocation, they just change places. So they're in different parts of the genome now, they're on different chromosomes, but phylogenetically, they're no different, right? From our perspective of just doing a gene tree, they look the same. It's when we start replacing 1 copy with the other through overriding or whatever you want to call it, that's when we start to see changes. So for example, if we have homeologous exchange where we replace subgenome A, subgenome B with subgenome A, then all of a sudden we've still have 2 copies. We still have 2 paralogs, but they've gone from allopolyploid paralogs to autopolyploid paralogs, right? So they're coming from the same ancestral genome and subgenome now, as opposed to 2 separate ones. And if we look at them in the phylogeny, we just have those 2 copies that we find in our polyploid now related to A. And we can flip the script and see the same thing if we do it with B, right? So a question would be, how often is this occurring? Because if this is occurring a lot, then it's a problem. It's like an interpretation of this could be problematic. So that's something we need to start to try to figure out, like how much of an occurrence is this, if it is indeed occurring. And we have, you know, good suspicion it is occurring. We see it across multiple lineages and plants. Yeah, this is something that happens after polyploidy, but what's its impact? So we've been working on this maize polyploid event for a while. I just started this postdoc with Toby Kellogg, and my lab has continued this since I started at UA. So maize is a tetraploid. It's an ancient tetraploid, somewhere between 8 to 12 million years. Take your pick. We've known about this for a long time, from like really cool work from like the early 1900s, looking at chromosomes and stuff like that, to the late 1900s, where people were like, yeah, it looks like a segmental allopolyploid based on these type of things, all the way up to Patrick Schnabel's paper, where they sequenced the genome and said, look at this. This is like your perfect little circle plot, right, of showing, yeah, there's a polyploid event here. There's a paper by James Schnabel, so different Schnabel, in 2011, where he started to look at fractionation across the maize subgenome. He kind of broke the maize subgenome down into pieces, like maize 1 and maize 2, based on fractionation rate. And this is an image, I believe this is for supplemental material of that paper. This is kind of like where he kind of envisioned the genome looking like at the time, based on the data that he had. And now this idea of maize 1 and maize 2 has been used by a lot of different people to kind of just ask these questions about methylation, expression variation, fractionation, all kinds of questions, because it's a good starting point, right? It's functionally, they seem to be different from each other. This is Rennie Byfield et al. In 2017. And so what they were looking at was, okay, if we look at maize 1 and maize 2 and we look at heritability of traits based on SNPs, what's the contribution between maize 1 and maize 2? They basically found across the board, for the most part, except kind of in a couple places, maize 1 seemed to be the major contributor to any given trait that they looked at. When they looked at other aspects of this, they found that it seemed to be paralogs that were more highly expressed explained heritability a bit more. And then also maize 2, which is more lowly expressed, had higher side-to-C methylation, so DNA methylation. So we're looking at CH, CHG, and CPG, upstream transcription start site. So it makes sense, like methylate and silence maize 2 over with maize 1 expressed. So in 2014, published a paper, this is out of Toby's lab with Matt Estep at the helm. I basically saying, okay, well, if we look at a bunch of different members of the Edinburgh, which is a tribe that maize is a part of, where are the polyploid events occurring? And what we found in that is that there is the polyploid event is shared between Z and Trypsica. So what this basically means is that all Zia and all trypsicum end up sharing this polyploid event. We since confirmed this using other types of data like transcriptomes. We then took that data and so that data is like a Sanger-based analysis, and we did a lot more species, pulling in lots of onless things. So maize doesn't have ons, but there's a lot of oned andropogony. So we thought, well, if we include all these onless things, maybe we'll start to see a pattern. And what we saw was, if we look at maize subgenome A and maize subgenome B, or 1 or 2, whatever you want to call them, one of these subgenomes definitely seem to be more closely related to some extant lineage. So this is some Eastern Hemisphere, African, Asian set of grasses in the Andropogony. You can see that their support values are like a posterior bit probability of 1, and the bootstrap values in the 80s. They're okay. I mean, better than some of these other values, especially like this, these lower values here, maybe for the this other onless clade being closely related to one of these other subgenomes. In all honesty, when you look at nuclear genes and do a phylogeny with Andropogony, the bootstrap values and the backbone of the group are always pretty low no matter how you cut it. So keep that in mind. So one thing what we wanted to do is we did like a whole chloroplast genome phylogeny, trying to get as many genera as we could, basically asking if it is a polyploid event and one of the lineages extant is the maternal donor lineage around. And what we found is it doesn't seem that way. There was nothing that was like sister to Tetraploid and Zia, suggesting an extant lineage that donated the cytoplasm to this polyploid event. This other group that we're kind of interested in showed up here, so it's actually not sister, it's kind of a little far away. If we take like a simplified chloroplast phylogeny, basically we get Zia and Trypsicum sister to each other, what we expect. Sorghum and Bossia, which is going to represent this Eastern Hemisphere clade, sister to each other. And then Soteria being our outgroup as another genome that's available. So we used a bunch of transcriptomes, and we've I started asking this question using more like a whole genome approach. And in part on this project, and in part on this actually Strawberry with Patrick Edger, I've developed this software called PhyDS, basically trying to identify like subgenome identity, like who's related to who and stuff like that. It's just an accounting thing. It's like really fast. And the idea is here is that instead of looking at a bunch of trees, we do it ourselves. And there's always room for improvement. I think there could be some cool likelihood stuff that could be mapped into this, but honestly, it's not something I have time to work on right now. But the general idea is like we give it a paralog set that we use from Synteny. So in case I know these two maize copies are related to each other, and I asked, well, are they more closely related to each other? Or are they more closely related to something else that doesn't share this polyploid event? So if you kind of walk back from this blue one, you'll see it's a bunch of Z and a bunch of Trypsicum. We know these all share that polyploid event, so that's expected. If we go to this other copy, you'll notice that there's no other Z and Trypsicum here, but there's Bossia and Eurolytrum. These are this clade of interest, this green clade. So we record the bootstrap value here as like 62% of the bootstrap trees supporting this relationship, and we just kind of put it out there. And we do this like across all our paralog pairs, across all these different gene phylogenies, and we get something like this where as your bootstraps decrease, you see more genes that support this, as you would expect. So if we kind of look in this area of actual decent support, you see a lot of cases where Trypsicums and Zia copies are just sister to each other, which would suggest that the genome, the extant lineages are not around. And we see a bunch of cases where this other lineage is also sister, but in way fewer number. So what we thought about that was, well, what happens if we just sequence the genome of like one of these things? So we're going to sequence the genome of Bossia. So we've been doing this with nanopores. We have almost 80x coverage in nanopore. We have the 60x coverage of Illumina Shotgun, a bunch of high C data. The short story for that is basically what we found is Bossia is an autopolyploid, and that long read from nanopore, it doesn't matter how much you have, it still creates not great assemblies. So we're actually partnering with Alex Harkis at Hudson Alpha to do a bunch of PacBio hi-fi now, and we think that's going to help us get past this point. We've had really great success using nanopore combined with high C and Illumina and other genomes, but this one's been problematic. So to get this quick question, I'll wrap this up pretty quickly. What about this whole idea of how is homeologous exchange affecting this? So if we come in and we ask if Zia paralogs are sister to each other when we use whole genome sequence, so we're looking for something like this phylogeny here. So if we look at 5,800 paralog pairs defined by Synteny, we find that of those pairs, about half of them, so that's paralogs, not pairs there, had bootstrap values at 50 or more when we looked at the phylogeny. So we see that about 2,600 instances of this phylogeny here, suggesting that Zia copies are sister to each other. Maybe there's no extant subgenome donor lineage or anything like that. When we kind of paste these around the genome, we get some pattern that looks like this, suggesting that there's some areas that are definitely more like this, but there's more variation across the genome than maybe originally would have expected based on some of our fractionation patterns. We can then ask the question, is there one of the subgenomes more closely related to this green group that we're interested in? In this case, we see about 2,000 of them, so a sizable number, about almost half of all the things that we looked at. But we also looked at, there's about 1,200 where it's an instance of a copy that's sister to a combination of sorghum, vassia, and the other paralog. So we're seeing it like basically trying to identify this individual and this individual. And we see lots of cases of this across our phylogenies. Again, when we look at these across the chromosomes, we see a certain pattern that pops out. If you compare them, there's actually some differentiation between them, suggesting that there is some variation across the genome of the signal that we're getting. This is another one of that. So are there instances where this can be explained by homoeologous exchange? So what if this pattern here isn't this? What if the pattern when these are sister to each other is actually vassia is like sister to them as a total, as a pair? So this would suggest there was homoeologous exchange with replacement. So we actually only see 428 instances of this pattern right here, suggesting that this might be present if it is in case, because we're assuming that vassia lineage contributed to subgenome, but it's a pretty low amount. It's like a quarter of like the total, or quarter to a fifth of what we see. So there's still some room for interpretation. I think as we improve the genome, we're going to get a little better with this because a lot of this has still got some missing data and things like that. But we'll see how it kind of as it goes over time. All right, so quick conclusion. Homoeologous exchange with replacement should be something you're considering if you're looking at polyploid phylogenomics. It's definitely there, and the question still remains, how prominent is it? And as we continue with the vassia genome, I think we're gonna hopefully be able to, at least for maize, start to ask that a little better. And that's all I got.
All right, thank you, Michael. That was great. Let's we need to get like an applause sound effect so that we all don't have to unmute. At any rate,
You can do an applause reaction.
oh, there you go.
Yeah.
Yeah. Well, I'm going to let everyone open the chat up again like we did last time. And I have to actually run here in about a minute for a faculty meeting, but I'm going to keep this Zoom going, and it'll be going on for actually another hour if folks want to stick around and talk to each other and ask questions and chat. Unfortunately, I'll be talking about furloughs. So anyway, I'll let folks open the questions up here in the chat block and let Michael handle those. So, all right. Thank you for a great talk. That's really cool.
Thanks. Sorry, I didn't see your warning time thing.
Oh, yeah, I've got to figure out the warning times without jumping in and interrupting audio.
Maybe you should just hold up a sign and label
yeah,
it.
exactly.
All right. So one of the questions was, do you mind sending me the reference that was about the difference between the two subgenomes of maize? Sure, Hannah. Which I'm not sure exactly which one you want, but I'm happy to send all of the references from the talk. So. All right, so Joel's asking, do you have a sense of how long homoeologous exchange tracks are expected to be, and trying to distinguish between those and gene conversion? It's not easy. You know, I think it's a great question. To me, it falls in that whole line, similar thinking of like, you know, what's centenic, right? So microcentenary versus like macrocentenary, and how big of these tracks we would expect to be. I actually don't think we have a sense of how big they would be. You know, we use these examples of like 4 genes and 5 genes or something like that, but it might be small, it might be large. One thing that my grad students actually working on as part of his dissertation is to ask this question in maze and to see basically if we think we have an instance of homoeologous exchange with replacement, and so we would expect that in those syntenic regions to see changes in signal. And so he's kind of looking at like a sliding window approach to look at variation in phylogenomic signal as he goes across those regions. That would allow us to maybe get to your question of like, what's the scale at which we're talking about here? If that makes sense. Let's see, Robin, do you know the timing of the Vossier whole genome enclosure event? It's also fractionating. The event is young-ish, right? It's, let's see, Vossier itself split off probably a few million years ago, so it's at least within then. So yeah, it's definitely fractionating, I'm sure. You know, it's fractionating, but again, it's autopolyploid. So, you know, it's still, those copies look so similar to each other. So the issue that I kind of mentioned before, and one of my questions for Kamil, I asked in his chat was basically, what do we do? Can we use like nanopore data kind of in that whole thing in GenomeScope or a Smudgeplot? Because basically the error rate of the nanopore reads pre-corrected is higher than the heterozygosity between the autopolyploid copies, the homoeologous copies. So everything's kind of swept up, and it's kind of capping us out at an N50 of like 200 KB or something like that right now for our assembly, despite throwing a lot of different things at it. But again, I think that this hi-fi thing will kind of get around that. So is there another polyploid event even predating the most recent common ancestor of maize and sorghum? Does it affect your identification homoeologous exchange segments? So as far as we know, the next event back in line is actually the row event, which is associated with all grasses. So that's about 70+ million years. That one's pretty well defined and pretty easy to see within the genome. So I actually don't think that one is causing a problem. There might be there's this weird signal that kind of comes up in KS plots and stuff when you look at the Edinburgh that might be a hybridization thing, but there doesn't seem to be any other pattern of polyploidy kind of catching up there. So Mike asked, how important is it having the breakpoints for distinguishing gene conversion, et cetera? That's a good question. That's what kind of like, I hope that Santosh can kind of start to ask that question with some of his work. You know, when we get a chromosome level scaffolded, at least Vossier genome, I think we'll be able to compare to maize like way more tightly. You know, Matt Hufford and his group just released all these NAM genomes, so it's like 25 new like awesome genomes for maize to play around with too, so we can really like look across lots of different lines and ask these questions. Can
I ask, I'll insert with audio real quick too, just a quick follow-up. How big are a lot of these homoeologous exchanges that you're seeing? I'm kind of curious, you know, how these compare to other sorts of
yeah.
You know, genome.
So I haven't actually gone back and looked at like chained the genes together to see what does the pattern look like. Right now, I think that the sparseness of the Vossier genome, because the Busco value is still really low, it's like in the 70s, is still preventing us from being able to do that well. My memory escapes me, but I'm pretty sure like in a Mimulus luteus genome paper and the strawberry genome paper, I think we might have some better sense of this. I know that like I did this a little bit with strawberry, and it's again, using the genes as the markers, it's, you know, that's kind of the problem, right? So it's just however close the genes are. Let's see, Paul, does your method for looking at homoeologous exchange consider incomplete lineage sorting? It does not, and it needs to. I think that's absolutely 100%. It was something I was going to mention as like my own caveat at the end, and I totally forgot. But Paul, if you want to talk about that, I would love to chat about that. I think it's be important to talk about that. Is categorizing maze 1 and maze 2 based on fractionation patterns still appropriate? I mean, the fractionation patterns don't change. There's still different fractionation patterns, and we still see patterns that kind of pop out with that. So far, what we're seeing with the evolution, like the evolutionary history, is that it's not perfect, but it's actually not bad. So in the sense that a lot of what maze 1 and maze 2 was originally called do represent different subgenomes historically, though there is more variation across them than originally expected based on, you know, these, in all honesty, very broad categorization of syntenic regions. And that's going to happen no matter what you do. The smaller you kind of break things up into microsyntenic regions, the more variation you're going to see. You know, because remember, synteny is part artistry as much as it is science, because it's all about changing the chaining distances and stuff like that. Was one subgenome dominant among all genotypes of particular allopolyploid? No, and one subgenome is not dominant across the whole either. Not always, not often. There's a lot of variation on dominance. You know, subgenome dominance is essentially the accumulation of site-by-site dominance to say this one is dominant over the other. You can have like 51%, or you could have 99% or something like that. But it varies a lot, and I think there's variation across. There can be variation across like lines and things like that too. Cool. I think that's all the questions for me. So.
All right, well, thank you everyone, and thank you, Michael. I'm gonna stop recording, and then I'm gonna have to leave the meeting, but folks can stick around and chat, breakout rooms and whatnot. I'll post a recording of this later on so that everybody can watch this later. And if there are any slides, I see that Kamil put a link to his slides there. Feel free to share those with me, but that's certainly not necessary. But I will post any links up on the website later this afternoon once everything's available. All right, everyone have it's a good beginning of the afternoon for us here. Have a good evening and a good day wherever you are. I have to get going, but I'll let my co-host Paul Blishak keep managing this. And again, thank you to Michael and Kamil for the wonderful talks. This was awesome. Thank you. Bye.